Welcome to our Wednesday Yachting Luncheon. Fun to be meeting live again, holy moly. Fun to see everybody here. So we're all humans. We've been humans for all this time, a million or so years. Who knows exactly when we should draw the line between when we became humans and when we were a previous previous living creature. But for how long have we dominated the earth? Just a blink in the history of the earth. In fact, even a smaller blink in the history of life on earth. How did life start on earth? Our speaker today delves into that topic in genuine scientific rigor. It's her field of study, and we're going to learn all about that. So starting in India, she was born in Delhi and raised in Calcutta, and got a degree in botany over there in India. Then she came to the United States and got a PhD at Cornell in biochemistry. She went back to, did postdoc work at Davis and UC Berkeley on biochemistry. She went to India, did a startup for a while, and then came back to America, working with Carnegie Science. Then she got a Rockefeller Fellowship, and now she's at Rockefeller Science at Stanford. So talk about a lot of big, wonderful names supporting her work. And so when we look at that sort of line of when life begins, the question that she's asking constantly is, what does it take to create life? This is particularly interesting when we look at what it took to create life on Earth, because one wonders, what does it take to create life on Mars or somewhere else? Some of the beginnings of what we think of as life don't really look like what we think of as animals and plants. But as she will point out, for most of the history of life on Earth, these little teeny microbes, which are now inside of our stomach, and a big piece of living organisms on Earth came from these microscopic little microbes that she has been studying and learning all about. Without them, there wouldn't be an us. So with that, I want us all to welcome up from Stanford, Devaki Bhaya, and come on up and spend some time with us and tell us about the beginnings of life on Earth. Yes, perfect.
Well, hello everybody, and is this good?
Yep.
Yeah. Well, hello everybody. It's a real pleasure to be here. This was done at somewhat short notice, but I jumped at the offer because it seemed such a remarkable group of people to talk to. So what I'm going to try to do in the next half an hour is I'm going to take you to two walks. One walk backward in time and one forward in time. And throughout that walk, we're going to be talking about microbes, which I love and admire, and I hope at the end of this talk, so will you. And what I'm going to try and tell you today is that, and I'll go on to the next slide here. So this is just to get situated closer. So this is just to get you, is this better? Just to get you situated, this is the dish at Stanford in springtime. You see oaks, you see mustard, oaks, all the gorgeous things that spring brings. And I am sure that you're not thinking that this place is teeming with microbes. And the reason you can't is on the next slide. We have an incredibly anthropomorphic view of the world. Everything is things we see. But down here in the tree of life, at the very bottom here, this little blob, can you see it? This little blob is all I'm going to be talking to you about. It's where life originated. So there's that first message is that life, all of this that we see and love and admire, could not have started in this tree of life without these guys. And so my job here is to explain to you why it's important, why it was important. So I like to think of this, that all of life has been punctuated by catastrophic events. So if we start back here, this is where life originated about 4.5 billion years ago. But very quickly, at about 3.7 billion years ago, we already have, and I will save you all the terminology, prokaryotes. These are simple cells. They had already formed on Earth. And these are there's one word to remember, it's the word cyanobacteria. Cyano means blue-green and bacteria. These guys were there almost 2.7 billion years ago. And they invented something that I'll come to in a minute. And so what they did was they changed the planet because they started evolving oxygen. Before that, all of this life was basically without oxygen. So a catastrophic event for everybody who lived there without oxygen. And what happened then is once these guys came along, and I'm going to show you what they did, there was a poll at one time saying, what are life's 10 greatest inventions? Here they are, even closer?
Yes.
Oh, wow, okay. Now I feel like a singer. So all of life's inventions are here, and the ones in green are the ones I'm going to point out to you. All of these are done by these cyanobacteria. They do photosynthesis, they're multicellular, they're a superorganism because they can do so many different biologies. And I've just shown you what some of those cells look like in real life. They are actually chains of cells, they can be single cells, and each cell is doing photosynthesis and producing oxygen. Produced oxygen over 1 million or more years till the levels of oxygen rose, and then other guys could start living. So if I'm not a yoga teacher, but I might have asked you to close your eyes and take a deep breath. That oxygen you breathe comes from bacteria and now comes from plants, which I think is something we don't appreciate every day. So it's a laundry list. I'm not going to bore you with this, but for some of you, it might be really interesting when they originated, which was over 2 billion years ago, like I told you. And because they can do photosynthesis, they can also fix nitrogen from the air. They become, if you think about it, they're really valuable partners. So a lot of other organisms just engulf them. And that's how we have plants today. Early organisms engulfed the cyanobacteria, which gave them all these new properties. And in a way, I think that has led the course of evolution of life on Earth. Okay? Now, this is what Ron was referring to, and this is a quote I take from Rita Colwell, who is the director of the National Science Foundation, and she said, "The general agreement is that 80% of the natural history of life on Earth is exclusively the history of microbial communities." So if you dwell on that for 10 seconds, it really behooves us to understand how microbial communities form, how they devolve, and how we can potentially use them. And that is the golden age of microbiology we are at now, that we can understand these microbes and we can begin to manipulate them. And that's what fascinates me about them. So if we were to take this walk backwards that I was talking about to you a few minutes ago, how are we going to do this? Can we take a walk backwards into areas that look like what early Earth may have looked like? And can we then begin to find things and life and then dissect them to understand what they're doing? Okay, does that make sense? So where did we go? We have a place like that. Remarkably, right here in the US, it's Yellowstone National Park, which has a number of these calderas that have formed over many years. So here, just to orient you, here's Wyoming, here are the calderas that form. And each of these, by the way, this is something that was seen in the Hunga eruption a few years ago, taken from NASA. Yellowstone was 100 times bigger than this. So this is another kind of catastrophe that happens in the Earth's evolution that changes the course of evolution. And so one might take an educated guess, that if you went to these calderas, if you went to the hot springs, we could go as close as we can to those early origins of life. And so the question that we started to answer many years ago, this is a group of us that went to Yellowstone and started this project funded by the National Science Foundation. The simple question was, how do microbes evolve and survive these incredibly stressful conditions? High temperature, high pressure, low pH. There can be very high sun one day, no sun the other day. And these guys, just to tell you, a microbe is about 50th or 70th the diameter of a single human hair. They're that tiny. And yet they're doing these incredibly sophisticated biochemical actions that have formed our Earth. So I just wanted to say that the Carnegie, I only found this out when I started working on them, that for years, this is 1935, this is years ago, got an interest in understanding Yellowstone hot springs. And that interest has continued not only at Carnegie, but around the world. Many people have realized that there's an incredible load of information and biology in these springs. So I'm going to take you to the scene of the crime. This is where we worked for many, many years. It's called Octopus Spring. And what you're looking at here is water coming boiling out of the earth, right? It's at 90 or 100 degrees. And then as it cools, you can all see this color here, right? This color is basically microbes. Microbes that make pigments that protect them and that they can use. And you can see, you don't see any of them growing here. It's just way too hot. But as soon as the temperature gets down to about 70 degrees, that's about the temperature at which you drink coffee, just to give you a sense. And now about 50 degrees, you basically have only cyanobacteria living in communities with a few of their friends. Below this, you start to get plants and diatoms and other whole lineages of life. Above these two, remarkably, there's a group of organisms called the archaea that can survive at boiling temperatures. Just think about that. So this little patch is giving us this incredible view of what life can be. Now, the question really then is, how do we do this? I mean, I'm sure this is running through our brains. These are good questions. How do you get to them? And the way we do this, I'm going to give you a very short description of all of this. You can take a little piece out of the mat that is growing, those colored mats, size of my pinky, by the way, because you're not allowed to take more than a certain number of samples out of Yellowstone. You can basically then, and this is a fascinating thing that has happened, you can just grind up this little piece of mat, and you can get what, don't remember all the words, but I would call it a metagenome, a sort of blueprint of everybody who's living in the mat at that time. You can also, and this is what we're doing in the lab, you can also start to grow them. Now, if you think about it, you have to grow them at those high temperatures. You have to give them everything they need so that they can survive. And another marvel of technology is that you can take these single cells. So here's a scale. They're micron-sized. You can flow them through a flow cytometer, catch a single cell, and get its blueprint, its genome. So now, in a nutshell, in this one slide, I have told you that you can go from a complicated community like that to understanding how they grow, which means you can manipulate them, you can understand their genome, and you can understand how single cells are growing. So it's almost like the whole arc of what we want to do when we want to dissect a system. Now, there's another revolution that's happened in microbiology, other than the genomic revolution, which I alluded to, and that is the ability to do microscopy at the atomic level. So each of these is one of my beloved cyanobacteria growing here. And you can see here, these cells are dividing. They're actually all held together by another thing I've studied for a very long time, something called pili, which hold them together and allow them to move towards light. Not what I'm going to talk to you about today. But this resolution actually allows you to see a cell, and I'm not telling you other work that you can do, where you can actually see where individual proteins are within that cell. So you have just an amazing degree of resolution of what's happening in the microbial world. So one little thing, and this might be just too much after lunch. Enjoy your lunch if you want to think about something else. But But I totally love this, and it has fascinated me for 20 years. I have to share this with you. So what you're looking at here, and think of it just as a circle that's colored, the rainbow colors, just to give you where particular genes are. Now, if two organisms are very, very similar, where their genes are, position of genes, will be more or less the same, right? Would you kind of take a $5 bet on that? So that's what we expected when we looked at these two organisms, very, very similar, other than the fact that they grow at two different temperatures. The A guy loves the high temperature, the B prime guy loves the low temperature. But what you can see here, I hope, and I'm happy to explain this more, is that they don't look like their genes are all in the same order. They've been scrambled. And that completely fascinated me because it was the first view we had of genomes moving around so that each gene was in a different relative position. So immediately my eyes lit up and I said, well, why are they doing this? How are they doing this? And what's the implication for evolution? And we have answers for many of these today. So I'm not going to bore you with all of those details. I will just tell you a few things we've learned, and maybe an interesting way, because I'm sure, and certainly talking to Ron, I kind of know that the world is full of folks doing technology. So maybe you can think of biology as hardware and software, the software being the genomes. And what we've discovered, what I just told you, are that they're really mosaic genomes. They're sharing things, they're moving genes around. And why does that excite me so much? Because it used to be that we thought genes and genomes are kind of inviolable. It's like an algorithm you don't change. And we've and it's beginning to be true for many, many organisms. Genes are moving in and out. Technical term for that is horizontal gene transfer. They're actually moving around, not in a linear way, but across phyla, which blows all of evolution in some ways out of the ground a little bit. And as a consequence of that, you can suddenly get a new function coming into a microbe, which it never had before. And so it can now move into a new evolutionary niche, which it didn't have the opportunity to do. And a consequence of all of this is that the hardware, too, has to respond to this. And we know how that's happening by taking a biochemical approach to understanding how things are changing. Once you change the software, how is the hardware? We're not as far along in trying to really manipulate the hardware as we can do with sequencing genomes. We spent an ungodly sum of money sequencing these two genomes. You can now send a microbial genome, get it back in about a week for about $100. So anybody can sequence their microbiomes, people do. You can sequence individual genomes. So really a revolution. So that's what I've told you about the cyanobacteria. And you might think, well, life's pretty good then. They're doing what they have to do. They can grow, they can thrive in these extreme environments. But life's not that easy. And life's certainly not easy out in the real world. And what we've discovered, so this is a somewhat artistic view of what's going on, but let me orient you and tell you what's going on. So here's a cyanobacteria. Say, for example, here are those things I showed you on the electron micrograph, the pili coming out. These guys that look like space aliens are actually viruses, or what we call bacteriophage. So they're even 20 times smaller, or even more than that, than the host. These guys also carry DNA, and they inject it into the cell and use the cell's machinery to grow and reproduce. Now, how ridiculously complicated that is, I can't even begin to explain to you, because we are at a completely new frontier in understanding the roles of viruses. But they can also move DNA around. And the reason I bring it up is I'm sure every one of you in this audience has heard of gene editing and the CRISPR story. Much of it started here at Berkeley and elsewhere. And it actually, many of you may not know this, actually originated by understanding that bacteria have this incredibly sophisticated way of fighting back viruses. And I'll spend a minute telling you that, although there are hundreds of fabulous YouTube videos in real time showing you how this happens. What happens is the virus injects a little piece of its DNA, and the host says, aha, that's like a calling card. I'm going to remember you. And the next time a similar or exactly the same virus comes in, the host uses that calling card to recognize this new guy, recognize it, and then kills the DNA, end of show. So it's really like an immunity system like we have. We think we are extremely sophisticated in having immunity systems, which are really a form of memory. We remember a previous attack. Well, let me tell you, bacteria figured this out billions of years ago. So we're now at this whole new frontier of understanding viruses. We understand they don't only just come in DNA, as many of us know, they come as RNA, they come single-stranded, they come double-stranded. Anything you can think of, we were probably going to find. And I would leave you with that thought that this is a whole frontier we haven't even gotten to, but we now have the technology to deal with it. So another thing that I just again want to leave a little snippet with is the fact that we're not talking about just a few things. We're talking about bacteria in the order of 10 to the power 30. We're talking about viruses, which may be 10 times more. We're talking about the number of bacteria on a human is on this scale. So we're talking about enormous numbers in which diversity and this kind of gene exchange is taking place. And I might add, as well, many of you will know, that the word for saying something is huge is astronomical, right? Stars in the sky. I would propose that we should use something like vironomical or bacterionomical to explain the immense diversity that we really have no clue of. But my guess is in the next 50 years or so, this understanding of bacteria using artificial intelligence, etc., will give us these protein structures, will give us interactions, things we could only dream of even 20 years ago. So how are we going to actually use all this information? So I'm going to end with a story that has captivated me for a very long time. And what this is showing you is a map of the world, showing you places where too much fertilizer is used, places where too little fertilizer is used. Too little fertilizer means your yields aren't good enough. Too much fertilizer leads to this problem. The fertilizer is just put into farms in China, in India, for example, where when you get fertilizer, you put it in, it runs off into lakes, and it causes these immense algal blooms. It causes what I saw actually in Qingdao when there was a sailing competition going on there at the Olympics. So I did throw in, Ron, on your behalf, something about the Olympics. And you could actually see these masses of algae that humans were actually having to shunt out of there just so that the Olympics could be held. So it tells you the scale. Even though they're microscopic, when they get the right conditions, they really can bloom. The word is bloom. And you can see this, actually, this is in Venezuela. This is a picture taken from space, and all this green color is actually algae and cyanobacteria blooming when they get enough nutrients. So this is a major, major problem. It's been the holy grail. How do we solve using so much nitrogen fertilizer, which is both an energy expense and then an bioeconomic disaster? And one of the things that many groups are trying to do, we've made a stab at it. And I won't bore you with all the details here. The simple idea is, as I've told you, cyanobacteria can fix nitrogen. That's a pretty incredible job. They can take nitrogen from the air and make it into ammonia. Now, that's what the Haber-Bosch process does, but it uses enormous amounts of energy and inputs. So the holy grail for a long time has been, can we take this nitrogen-fixing capacity which bacteria have and plants do not, and put it into plants. And if we could, then we would bypass all the problems of having to feed fertilizer to plants and the downstream effects. It's still a bit of a dream, but we're taking bits and pieces, and that's why I was talking about the fact that we're now getting more of the pieces together to understand how we could transport this into plants. And you know, stay tuned. I think people are working on this. Many groups around the world are doing this, particularly in the US. And so the holy grail would be to put it into something like corn. So leave you with the question of where are we going? Where am I going next? And it's looking backward and looking forward, as Janus did. And it's thinking now that we have understood, and I've given you the lens of cyanobacteria. Hopefully that's one word you will remember after this talk. They're immensely diverse, immensely gifted, but they do not live alone. They live in communities. And I think the next future is trying to understand, what do they do in communities? Why is it so important for them to live in communities? And that's shown here. These are different bacteria that we grow in the lab, moving out in different ways when they're in communities. This is just an AI-generated figure, which I love to throw in because everybody can tell the real thing from the false. But that's where we are headed, trying to make communities where we can predict what the behaviors will be. We can manipulate those behaviors. And so I didn't want to end this at this esteemed place and not talk about the role of yachts. So some of you may have heard about the Tara Oceans project. Have any of you heard about that? So this is a project that was started, I believe, in France. I don't know all the details, but basically it was taking this yacht it has a very interesting history I can tell you more about. And it has traveled around the world taking samples of plankton, of algae, bringing them back to the lab and sequencing them, trying to grow them. It has been a revolution in understanding plankton. Plankton, by the meaning words wander, they go all around the ocean. We knew nothing about them. So this is the kind of thing that yachts can do. So let's not discount them. Let's also not forget that even though my talk was focused on the extreme environments of Yellowstone, you can go down deep into the oceans. These are the famous black smokers, and you see bacteria there. You see these incredible tube worms also doing a lot of their biochemistry and geochemical cycle through the use of bacteria. So there again, it's not just Yellowstone, it's not just in our guts, it's not just in the soil. Everywhere you look, if you care to look, there are bacteria doing their job. And so, you know, yachts may be one way to go after them. There may be others. So I'll end here, and I will end with one of my favorite quotes from Darwin, who, by the way, didn't know of the existence of bacteria for the same reasons we didn't. We can't see them. But he said, it's not the strongest of species that survive, nor the most intelligent, but the ones most responsive to change. And I think bacteria are the masterminds of being able to respond to change. And I think that that's just an incredible untapped resource that we have at our fingertips if we were to want to use them. And I'll just wind up by saying that I've been a member of the California Academy of Sciences right here in San Francisco. Don't get down here as much as I can from Palo Alto. But we did this right in the middle of the pandemic, as you can obviously tell. And we were showing the folks who came that Thursday evening how you grow algae. We did something called cyanotype, which I won't tell you all about, but really engaging the public in understanding science. And I've always enjoyed doing that, which is why when I got this invitation short to do this, I said, sure, I will. And I'll leave you with that picture and the Carnegie institution where I work, beautiful, venerable old building. This was a group picture taken after the postdocs organized a symposium. And that's where I'll end, and I'm very happy to take questions. And move over there, right?
Wonderful. Yeah. Devaki. Okay, so there are 8 billion people on Earth.
And growing.
And growing. And how many microbes in each of us?
So, like I said, something to the 10 to the power 14.
So give us that as, I saw 10 to the power 14, but I can't remember, is that quadrillion? What is to the power 14?
What? I wouldn't know what the terminology is, but
Okay.
something like that.
So,
right?
so what's
so several orders of magnitude more than we have,
okay,
right?
so we have
10
8 billion
10 to the
humans,
9, yeah.
and we might have 9 times that number of microbes in each one of us.
Individual bacteria.
Individual bacteria.
So that's more, but this is something I should clarify.
Okay.
It's debated whether it's 10 to the 14th, how did we overcalculate, undercalculate, but for sure it's very high. And something we didn't appreciate.
Now humans are growing. That is to say, our $8 billion population is going to keep growing. Like
maybe
2%. It's been tapering off, but some
Yeah.
areas are growing faster than
Yeah.
America, for instance.
Yeah.
But if we said 2% growth, is the number of microbes growing? Is it growing the same rate or slower than humans?
I would say much more.
Faster.
Much faster in the sense that it's an untapped thing. We look, again, talk about anthropomorphic, we look at our gut microbiome. We know more about our gut than we know about microbes anywhere in the world. And we know that even in the gut, I'll frame your question differently. I'll say that different groups of people, depending on their diet, depending on where they live, have different groups of bacteria in them. And when we fly from one continent to another, we are carrying bacteria with us. So this idea of constant dynamics in microbes is certainly happening. And it's huge.
So now humans live on average around the world around 70 years. America's older, but 70 years. What's the life cycle of a microbe?
I will ask you this question. If I take one cell and separate it into two.
That's it. That's the moment right there. Okay, so that's the birth of a cell.
That's the birth of a cell.
Okay.
In E. coli, which I'm sure all of you have heard of, that's the beloved organism we know, can divide every 20 minutes.
20 minutes.
Every 20 minutes you get a copy of the
Okay.
DNA, the protein. There are others that it's literally estimated to be thousands of years before they divide. So they just sit there. So, for example, in the black smokers, even in our hot springs, they just don't have the nutrients to divide, but they sit there.
So they're
waiting.
nutrient starved until they get to a certain point, and then they can divide.
When they get the nutrients.
When they get the nutrients, when they get the required nutrients.
Yeah.
So you're saying that some are in seconds and some are in...
minutes. Seconds, I wish. But
Okay.
it's minutes
Okay.
or years.
Now, insects, little ants that have
Yeah.
a short life cycle,
Yeah.
are there microbes inside their gut too?
Absolutely, and there's somebody at Caltech who looks at termites. Why termites? Because termites can actually eat wood. So you find bacteria that can actually break down the lignocellulose. So he went looking in termite gut specifically to get bacteria that can break down lignocellulose. And that's exactly the point I was making. Wherever you look, you see bacteria using the resources they have.
So now if I have my telescope, I look out at the sky, but if I have my microscope, can I actually see a microbe?
Yes, and if any of you would like to come, for
Stay
sure.
on the mic. Okay.
You can see them.
Okay, you can see them.
You can see them. You
Okay.
can see them move.
Okay.
You can see them divide. You
Okay.
can see them go through their life cycle.
Okay. And so when you see one divide, essentially a new one is born
Yeah,
in that
yeah.
moment, you can actually see it in the microscope. At what power are you looking at in the microscope?
So for the ones we look up, I can say at about 20x magnification
20x.
or 200 magnification.
Okay.
For sure you can see them. And you can go up to, in an electron microscope, to say something like 45,000, in which you can see individual subparts of a cell.
Now, I'm going to keep asking questions of our wizard here, but if you have a question, you can raise your hand. We have in our audience, Fritz Maytag, founder of Anchor Steam Beer, knows a little bit about bacteria. Johnny, would you give him the mic? Because I see Fritz has got a question. Is bacteria our friend? I'll ask him. It might not be the friend of a brewer. Fritz, you have a question.
I do. It's actually not about bacteria, although I'm with you 1,000%. I'm just intrigued by your stories. NASA is interested in finding whether there's life outside Earth. And I understand that NASA has a committee or a department that's trying to decide what is life. Because if they find something out there that's gooey and kind of has some of these attributes, is it really life? Do you have your own idea about how to define life?
So this is a debate that is fascinating and very important, and you hit the nail on the head, because we define life as we know it on Earth, right? So, for example, one of the things is if you see oxygen, that's life. If you see structure, that is life. But that is not necessarily the only definition of life. So people, in fact, I will send you this wonderful article that just came out last week in Nature, talking about the fact that we don't really have a paradigm for understanding even the definition of life. And I'll stray a little bit away, but it'll interest people. How many of you would say that viruses are life? With some of you, yes. And why so?
They reproduce. They reproduce, and they're individual, and they can change. They vary as they reproduce.
But they cannot live on their own. They have to have a host. Mm-hmm. So
they're
some
parasitic,
people would say that that's maybe not life. So it depends on what criteria you use. But the question you're asking, which is NASA's question, is there life somewhere else? And how do we detect it, right? Is a different story altogether. Because now you're saying, what are the parameters that we can confidently say must be a reproducing system? And to me, that is maybe the easiest way to think about it. Is it able to reproduce? But we can't actually see on Mars if it's reproducing. We have to see the output of that and be confident that that's it, right? The other argument that's often made is maybe there are certain chemicals that can only be formed by life as it exists on Earth. And I think we're extremely limited, I would say lacking in imagination in what life could be. But I give ourselves a pat on the back because we don't know how to think about life on other planets. And the last thing I'll say about that it is not a question that is resolved. And this article is really arguing for the fact that we need to have more groups of people sitting around a table and saying, how do we do this? We're too siloed. So the guys who do the geobiology think about it one way. The people who are just getting back Mars samples think another way. People like me who are biologists think about it another way, but we haven't stretched that question enough.
So about how do microbes create oxygen on Earth? How do they do it?
So it's not all microbes, by the way, it's just a few, the cyanobacteria. Mm-hmm. They actually split water. That's what photosynthesis is, which is why it's so incredible. It's taking something that's abundant, a source like water, and basically using a membrane and proteins to split water. And one of the outputs of water, of course, are electrons, which then drive an electron chain. But the other output is oxygen.
And so we, in our form of life, we're using oxygen. Much of living creatures use oxygen. Yeah. But that's only possible because of the work done by the microbes to make oxygen by splitting water.
Absolutely,
okay.
Yes.
So you're saying they also create the condition with which the forms of life we're familiar with can exist.
Evolve and exist.
Exactly.
But I should add that before oxygen was created by the splitting of water by cyanobacteria, there were what are called anaerobes. They lived without oxygen. And actually, interestingly, those guys have been driven away from most environments now. So you can find anaerobes, strict anaerobes, that will live in environments where there's no oxygen.
And they predated the oxygen requiring creatures. Okay.
Yes.
So now, I've been to Clear Lake, and it's anything but. Probably because of your microbes. it's your doing. All these microbes create the algae. I know it's your personal doing. They create these algae, and I went skiing on my boat as a young guy when they were beginning to bloom up, and I thought to myself, "What on earth?" Well, now of course we had a speaker a year or so ago talk to us about a bloom of algae that was in the Caribbean that went like 1,200 miles out into the Atlantic. 1,200 miles.
Yeah, you can see from space.
Exactly. Yeah. Mm-hmm. Okay, what on earth is going on that the microbes are, overwhelming us like that?
Well, and in fact, sorry?
Tinsley, too, exactly. John Harrington, our librarian, points out that at Tinsley Island, we've been overwhelmed at different times with hyacinth and sometimes all kinds of forms of microbes that create this green film on the water. It's our pristine water. How'd that film get there?
Because those guys were sitting there at very low levels, basically starving. The moment you give them nitrogen and phosphorus, aka fertilizer
runoff from the street,
from streams, from fields,
from agribusiness
golf
in the delta
courses, run into the lakes, and then you get these giant blooms. And so the real question...
So it's a chemical imbalance created by runoff, in these cases from fertilizer. That's what you're saying.
It's not necessarily imbalance. It's that they were waiting at the stasis point. Couldn't grow. Fertilizer comes, boom, in a week. I should also say that in California, we didn't have that many blooms. They're getting more and more, and one of the issues is it's probably due to rises in temperature.
Okay.
Right?
So rising temperature enables the microbes to grow
faster and more swiftly. So Clear Lake, actually, I should also mention, is a water source for many tribes that live around there. And so they're very concerned. They're actually taking a lot of initiative in measuring the levels of toxins from cyanobacteria. And there's a whole fantastic website giving you day to day the level of these toxins in the water, because it's really a crisis. Once you get a bloom and the toxins are released, there's not much you can do. So one of the things we've been thinking about in the lab a lot is, can we get biosensors that catch these blooms very early on and sort of get them before they've bloomed?
So I understand how bio runoff in the agri-centric delta could cause blooms in the delta. But 1,200 miles out in the Atlantic, what happened in the Atlantic that caused these blooms to grow and to kind of overwhelm this, animals on the surface, to cover the surface of the water with these blooms?
That's a really good question, and that is because there's upwellings of nutrients. The nutrients sink at certain times of the year. They can come up, and then that's when you get a huge bloom.
So now, how can we harvest these things that are growing so much?
To get rid of them or
how
use
can we
them?
Is there some value? Can we turn the problem into an opportunity?
So in the case of seaweeds, for sure, and many people are doing this. With microbes, I don't think there's any obvious way to harvest them, but there could be. We just haven't thought about it enough. Seaweeds grow incredibly fast, and I was talking to somebody in the audience today that it's an open opportunity to start using seaweeds in the oceans.
And so we've had some speakers who talk about the different types of seaweed that they're breeding and growing, which they feed to cows, and that reduces the negative methane output, burping, mostly burping, from the cows. And essentially that's a way of harvesting this sort of bio process of the seaweed being grown. And they're basically modifying genetically the seaweed and making seaweed that has a different effect on the inside of the cows.
And that's the kind of thing I was sort of alluding to, that if we know the problem, if we can manipulate these, then we have a chance at fixing the problem. Otherwise, it's just a runaway problem.
Okay. So now, what scares you?
How long do you want me to answer that question?
I want a short answer. All my answers must be short.
I think it is what I call catastrophes without borders. Pandemics, growth of microbes that we can't control, viruses that will run amok. I mean, biologically, what we were as humans, we were separated. We didn't really cross-fertilize that much. But now, as you know, with the SARS pandemic, for example, it was a virus moving from some kind of animal source to humans. That is going to happen more and more. Bacteria are going to grow more and more, and we don't have a good way of controlling them.
We can't control those little buggers. Darn it. Okay, so, but their chemistry, why can't we basically introduce a solution, a chemical of some kind, near them? I mean, after all, aren't we the gods of the universe? Can't we basically control these they're just little things?
Well, I'll tell you one thing that they're doing, and you tell me how great that is, right? So one of the things with these toxic blooms is if you put in hydrogen peroxide,
okay,
often that kills the bacteria.
Oh.
Right? How much hydrogen peroxide would you want to be pouring down into some lake, right?
Are there guns of hydrogen peroxide? No.
Catch each microbe.
Okay.
But so basically, I think my solution to that is not to catch it when the problem has gone too far. Mm-hmm. Get early warning signals that something is wrong.
Okay.
And catch it before you have to bring the big guns out to get them.
Now, how can we use this growing force of microbes to solve a bigger problem? We have this micro pollution problem in the ocean right now.
Microplastics.
Microplastics are getting smaller and smaller and smaller, and if we can stop them from running down the rivers in the Philippines and India, Pakistan, that would be a start. But there's a lot of them out there already. And can these little microbes at some point serve us by eating or getting nourishment from or somehow taking advantage of these small little microplastics, which are growing? Earlier speakers have told us that they're beginning to coat parts of the bottom of the ocean.
Yeah. They're coming everywhere, and we can't stop it.
Microplastics.
Microplastics. The question of using the microbes, there are lots, and so I'll raise an interesting point for this audience. One is there's a lot of science being done in labs, right? In fact, I was part of a grant that was trying to understand microplastics and can we degrade them using microbes.
Right, exactly.
Right? So you might get a little hint. This microbe can break down this kind of microplastic, right? But to go from there to scaling is a major issue, and it is a major issue with everything. I believe that we will find microbes that can break it, because in fact, many of you know this, when there are large oil spills, right, some tanker releases, 3 months later they said, where did it go? And they actually figure out that microbes have broken down the oil. So I truly do believe that plastics can be broken down. But everything, I think in my mind, I love the biology I do, and I'm interested in the sort of basic research, right? Scaling that to something that solves a problem, I think needs engineers and scalability.
So about a month ago, we had a speaker who has the biggest marine salvage business in the world. And he's timed up with the environmentalist Mary Crowley. And he basically described just what you said. He said, we're taking on these giant oil spills in old mechanical ways, but we're exploring ways to use bacteria and other organic microbes who could possibly eat these and break them down. And so what you're saying is, yes, you can imagine a future where we harness the power of these, ubiquitous little engines, the little bio-engines.
Little bio-engines, for sure, and also that they can do it in very large numbers. If you can think of, the way we produce drugs to scale. We don't make insulin. We don't get insulin from pigs anymore, right? We grow them in fermenters. That was one of the feats of genetic engineering. The real issue is the scale, but that I feel is not necessarily the biologist's domain. It's to give them the tools and say, well, is this scalable? Should this be scalable?
So you're saying you're not going to productize it. So we have a speaker. Bring the microphone, John, to the lady here. So you're saying that you, the biologist, the scientist, won't necessarily be productizing this technology, but you can keep discovering it, inventing the kernels of the technology that later can be productized.
Yeah, so you asked me what my nightmare was?
Yes.
I'll say what my dream is.
I wanted to get to that. Yes, what about your dream?
To have biotechnologists and
Stay on the mic, though. My dream is that you stay really close to the mic.
Not to kiss the mic. So my dream is to work both to have biologists, biotechnologists, and folks like you who say, this is not a feasible problem. This is something you can go to. But we don't talk to each other. And I think that is a grievous problem. The National Science Foundation has this whole new, I don't know, very big venture called the bioeconomy, which is basically to say we've got to use the tools of biology. But I would say we've got to work with the technologists, the engineers, early on.
Industry.
I believe that.
Here.
Question for you. If we manipulate microbes to solve a particular, perhaps man-created problem, are we unwittingly possibly creating yet another problem, particularly if it's done at scale?
Absolutely. And that is, I mean, talking about nightmares, that's the world's nightmare. It's... So I honestly feel that it's something we can just say we will not do, but I don't think humankind is set up for saying we will not do this. It will happen, there will be issues, and we will figure out ways to solve them. Cynically, I would say that that's going to happen.
So we're chasing our tail, basically.
It's somebody
to
else's
be cynical.
tail, maybe.
But isn't it really the nature of man that we keep solving these problems? That's how we got where we are right now. We keep inventing technology to solve a problem, and then another problem, and then another problem. So I see this as the opportunity, a giant opportunity. We have big problems, and now we've got these little bioengines, and how can we put these bioengines to work, helping us solve these big problems that we have?
Yeah, I mean, there's this whole field of what I didn't talk very much about, but what I was talking about with putting nitrogen fixation genes into corn is what's called synthetic biology. It's a huge new field. It alludes exactly to what you were saying. Once you start putting genes from one organism into another, have you created a Frankenstein you haven't realized? And there are both camps, obviously.
So now the CRISPR, we had a speaker talk about the CRISPR a couple years ago. And so genetic engineering using the tool of the CRISPR, how do you use a CRISPR to change the biology of these little microbes?
Oh, easily. So what you're basically doing, if you think of CRISPR as being a pair of scissors, but a pair of molecular scissors, you can go there. Let's say a gene is basically a string of letters, and if you change the A to a C, you have a problem. So can you go in with your molecular scissors, snip out the A, replace it with a C? That's what CRISPR does. Now, going back to your question, what has worried the field for a long time is what's called off-target. You're doing this, and have you inadvertently changed something else? And the technology, again, is getting much better, that those frequency of errors is going down. And as many of you know, there's, been examples now of gene editing for humans, getting rid of, dreadful genetic diseases.
So now these little microbes, they can live in amazingly tough conditions. And you're telling us that inside of Yellowstone's geyser, there are living little microbes that are right there in super hot water, close to boiling rate. And then they, when they land and they create these big stains, that's living matter changing color of the terrain. Those microbes there. So now, how long was the Earth like that?
On one of
3
my
billion
slides,
years?
It's 2 billion, I would
2
say.
billion
like
years or
that
so.
It was basically just microbes living in community, which is what the word I was telling you yesterday about stromatolites, are these ancient accretions of minerals and organisms that just grew on top of each other. So it was almost the space between geology and biology.
So it kind of looks like a chemical process, but really it's a biological process.
That's right. That's right. That's a good way to put it.
And so when we think about potential life on Mars, are you saying that it could be that there could be something like this that's growing in the inhospitable environment where there's low oxygen on Mars? What about that?
I would not hazard a guess on what that would look like. But people are starting to see, was there evidence of water? That's another thing we assume, that life needs.
Uh-huh.
And so if you have water, did you have the building blocks, for example, the amino acids, etc., to put together a cell?
Any more questions? I'm looking. Oh,
Yeah.
there's a question. Fritz has a question back there. So essentially, on Mars, we could have these kind of
Remnants, yeah.
okay. You could microbes could be living there without oxygen, but they could. You're giving me a frown. Could they? Could they live without oxygen? Yes.
Certain microbes
Yes.
can, we know
Yes.
that.
Yeah. Exactly.
Yeah. But
Okay.
did all those conditions happen again on Mars
Right.
that
Okay.
allowed it to happen here?
Fritz.
You mentioned water. I'm intrigued by the question of where our water came from. It used to be that many people thought it came from comets hitting the Earth with ice on them. Do you have an opinion about where all of our water came from?
Actually, I don't, and I wouldn't hazard a guess. But there has been, and I'm trying to recall the details of that, evidence that there's water in the core of the Earth. I can't. Is that what you
That
read?
seems to be the opinion seems to be moving in that direction.
And that's what I read, but that's, I would not hazard a guess. But it is assumed that life on Earth needed water.
Now, what ambitions do you have? Give me a dream. What do you wish for right now? You're a scientist. What do you wish for?
I have dreams in two different directions. One is the dream of science, which is to understand and manipulate communities so that we can really get them to do, not in an ad hoc fashion, this worked, I'm going home, but get it to do it reproducibly and to create communities that do something stably. That's one dream. The other dream I have is completely different, and that is the dream of education and science. Because I really feel that, it's the very small proportion of people who get to do the kind of science in the untrammeled way that we do it. But I would like for children to feel that science is something anybody can do. I grew up in India, and I was just fortunate, like I was telling you on the ride up here, that I had folks who cultivated the interest of science in me. I think we need a lot more of that. And again, I think about partnerships. Why shouldn't universities and institutions do this as part of the training of postdocs, of graduate students? I think it would work both ways.
So now you also have kind of an idea of a bioscience museum.
I do.
My son-in-law is in the Rock and Roll Hall of Fame. It's a kind of museum for rock and roll, and there's museums for old cars. There's museums of all sorts.
All sorts.
Fritz is even in the Smithsonian Museum. But what about your museum idea for a bioengineering museum at Stanford? Talk about that, please.
I'll talk about it as long as all of you never repeat this to anybody, because this is still in my head.
And given that this is being live streamed on YouTube, don't worry about anybody hearing it all.
Yeah, so somewhat tongue-in-cheek. But the fact is that Stanford, as you know, is technology, on steroids. And yet it has two fantastic art museums. Many of you, I'm sure, have been there many times. It doesn't have a science museum. And suddenly it hit me like a rock. How is that possible? Why don't we have streams of people coming through to Stanford, children coming through, and say this was the birth of hundreds of inventions. And one thing I was thinking about is, can we carve a little space on the campus where we can show these kinds of experiments, have it as what I would call living science. It's not science in a textbook. It's not somebody, in a white coat sitting somewhere. It's people doing science. And I got this idea years ago, since you asked, at the California Academy, where they actually have scientists who are cleaning up bones and fossils. And the children love that, because suddenly they see this is how it's done. It's not done behind closed doors that nobody sees. It's actually curiosity. And a slide I usually put up, I forgot to put up here, fantastic quote by Dorothy Parker that says, the cure for boredom is curiosity. There is no cure for curiosity. And I love that, because I think that is emblematic of how scientists think. I get up with 100 different questions. I solve maybe one, but that is the mind working, right?
Absolutely. Devaki, Bhaya, Senior Staff Scientist, Carnegie Science at Stanford, thanks so much for sharing your insights with the
Thank
Wednesday
you.
Yachting Luncheon. Yay, good on you. Thank you. Much fun. Perfect. And with that, we adjourn the luncheon.
All right, thank you.
This has been a presentation of the Wednesday Yachting Luncheon.